Hello, everyone, and thank you for coming to my talk. Let me just share my screen. All right, hope this is working. So first, I'd like to thank Olivier and the organizers for the opportunity to speak, and more broadly for putting together this really wonderful conference during such a challenging time. Today I'd like to share with you our work on the structure, biogenesis, and engineering of the eukaryotic CO2 concentrating organelle, the pirenoide, which is found in nearly every eukaryotic alga and plays a major role in the global carbon cycle. A pirenoide is shown here in this beautiful electron micrograph from Robin Roth and Ursula Goodenough. So our story today begins with my favorite enzyme, rubisco. Rubisco is thought to be the most abundant enzyme on the planet, and it catalyzes this key reaction of fixing CO2 into biomass in essentially all photosynthetic organisms. Rubisco is very slow, and the slow rate of CO2 fixation limits the growth rates of many of our crops, including rice and wheat. Now, we've known about this issue for many decades now, and whole careers have been spent on trying to engineer a better rubisco. Nature has been working on the same problem for much longer than we have, and neither we nor nature have been able to produce a better rubisco. However, nature has found a different solution to this problem. It turns out that some organisms have evolved a way to make rubisco run faster by putting rubisco into a compartment into which they pump CO2 to a high concentration. This is called a CO2 concentrating mechanism. Eukaryotic algae, prokaryotic cyanobacteria, and a few land plants called C4 plants have each independently evolved a different molecular way to achieve such a CO2 concentrating mechanism. And there's currently a great interest in understanding how they work and in transferring them into the crops that don't have them. A lot of solid science suggests that if successful, this transfer could increase yields by up to 60%, and the crops would require less water and less nitrogen fertilizer. Now, it's early days, and I think at this time all of these approaches are worth pursuing, and my lab in particular has focused on understanding the algal CO2 concentrating mechanism and transferring it to land plants. And we've done this as part of a wonderful collaboration called Combining Algal and Plant Photosynthesis, which started 11 years ago as a collaboration between my lab and two British labs, those of Howard Griffiths and Alison Smith. And over the years, we've added many wonderful collaborators, including Alistair McCormick, Luke Mackinder, Mark Stitt, Ursula Goodenough, Ned Wingreen, and Ben Engel. To help you understand what we're doing, I first want to explain the major difference between CO2 assimilation in algae and CO2 assimilation in plants. In both algae and plants, the CO2 assimilating enzyme rubisco is found inside the photosynthetic organelle, the chloroplast. In a land plant, the rubisco is found throughout the chloroplast, and CO2 diffuses in slowly from outside to be assimilated by rubisco. In contrast, in an algal cell, the rubisco is clustered into a microcompartment called the pirenoide. And CO2 is pumped from a low concentration outside the cell to a high concentration inside the pirenoide, which makes the rubisco run faster. Now I want to show you what this looks like in an actual cell. This is a cross-section through a single cell of our model alga Chlamydomonas. I've highlighted the pirenoid in teal and the surrounding chloroplast in magenta. It turns out that having a pirenoid confers such a tremendous growth advantage that nearly all algae on the planet have one, including the algae that do much of the photosynthesis in the oceans: diatoms, coccolithophores, and chlorophytes. In fact, we calculate that about 35% of global CO2 assimilation occurs in a pirenoid. So this is clearly an organelle of major biogeochemical importance. And yet it's also one of the most poorly characterized organelles that we know of. If we zoom in on the pirenoid by electron microscopy, we can see at least three subcompartments. there's a spheroidal matrix containing tightly packed rubisco, which I've highlighted in teal. The little clumps you see here are actually individual rubisco holoenzymes. The matrix is surrounded not by a membrane, but by a sheath made of starch, which is a polymer of sugar. And the matrix is traversed by membrane tubules, which exit through cracks in the starch sheath and merge with the photosynthetic thylakoid membranes outside the pirenoid. And these tubules are thought to deliver concentrated CO2 to the rubisco in the matrix. And until very recently, this has essentially been the extent of our understanding of the pirenoid. This fascinating cellular structure has remained almost completely uncharacterized at a molecular level. But if we dream of engineering a pirenoid into land plants, we need to address a number of basic questions, like what are the protein building blocks of the structure, and how is it assembled? Today, I want to share with you some progress that we've made on these questions over the past 11 years, which brings me to my outline. I've just told you that the algal pirenoid enhances CO2 fixation. I'll tell you how we developed new tools, including a mutant resource, to discover new components of the pirenoid with high-throughput genetics. I'll tell you how we used a systematic localization strategy to understand the anatomy of the pirenoid. I'll share with you our surprising discovery that the pirenoid is a phase-separated liquid-like organelle and not a crystalline solid structure as it had long been thought to be. Then I'll talk about our molecular studies of the pirenoid, which include the discovery of a key linker protein that holds the rubisco together in the pirenoid, and our discovery that rubisco binding motifs target proteins to the pirenoid and hold together the pirenoid's 3 subcompartments. Finally, I'll briefly mention some early progress in our collaboration's efforts to engineer a pirenoid into land plants. One of the biggest challenges that we faced when we first started was that we knew almost none of the protein components of the pirenoid. We reasoned that we could make rapid progress on this question with yeast high-throughput genetics. All we'd have to do is screen the mutant library, determine the localizations of hits, and we could even measure genetic interactions to place the new components into genetic pathways. The only problem with this idea is that yeast doesn't do photosynthesis, and it doesn't have a pirenoid. So we decided to develop these kinds of tools for an organism that does, the model eukaryotic alga Chlamydomonas, which we in the field affectionately call clammy. Clammy is the leading single-celled model organism for molecular studies of eukaryotic photosynthesis. And it's also made major contributions to our understanding of cilia biology, interorganelle communication, lipid metabolism, and the channel rhodopsins widely used in optogenetics come from Chlamydomonas. Like yeast, clammy grows as a haploid, which facilitates certain genetic studies. But when we started, progress in the hundreds of labs using this model organism was greatly limited by difficulties with obtaining mutants in genes of interest. No mutant library was available. The genome is 10 times larger than that of yeast. There was no effective nuclear gene targeting. Transforming DNA integrates at random sites in the genome, and mapping of these random insertion sites was mysteriously hard. We later figured out that this is because clammy chews up the foreign DNA, which leads to very messy insertion sites. And propagation was very low throughput. Cultures were propagated manually in individual glass vials, as you can see here. To overcome these challenges, we started a massive team effort. Led by 2 outstanding postdocs, Xiaobo Li and Ru Zhang, who are now both running their own labs, with our bioinformatician, Veronica, and the help of 6 technicians, and a lot of advice from our collaborators, Pete Lefevre, who runs the Chlamydomonas Resource Center, and Arthur Grossman, who was our next-door neighbor at Carnegie, where this project started. And together, we overcame the challenges that I mentioned and developed a genome-wide collection of 60,000 clammy mutants with known gene disruptions. This is a picture of several plates from our collection. In each of these colonies, a different gene is disrupted. 80% of the genes are represented. This is the first such resource in any single-celled photosynthetic organism. Over 3,000 of these mutants have been distributed to over 300 laboratories worldwide, and this resource is having a transformative impact on all the fields that use Chlamydomonas as a model system. The mutants can be searched on the website chlamylibrary.org, which also contains a large number of mutant phenotypes that we obtained by high-throughput genetics. And we'll soon also have high-throughput protein localization and protein-protein interaction data. So I encourage you to search this website for mutants in your favorite genes, and to also consider using this resource for your research. Now, one of the first things that my lab did with this resource is a massive screen to identify mutants with defective CO2 concentrating mechanisms. Leif Pallesen, a postdoc in the lab, grew each plate of mutants under two conditions, air and high CO2. He then looked for mutants like this colony in the middle of the zoom, which cannot grow photosynthetically under air, but can grow under high CO2. This classic phenotype suggests a defect in the CO2 concentrating mechanism. The screen yielded a list of parts, and we were pleased to see that 4 out of 6 previously known genes were hits. And we were thrilled to see that we had additionally discovered dozens of novel candidates. The next challenge was to figure out what these candidates are doing. Luke Mackinder, who at the time was a postdoc in my lab and who now runs his own lab in England, reasoned that a good way to identify and characterize pirenoid components was to start with a set of candidate proteins and then determine where each of these proteins localizes within the cell. However, at the time, it was very difficult to determine the localization of even a single protein in algae. So Luke developed a pipeline that allowed him to tag over 100 candidate components with the fluorescent protein Venus, transform them into chlamyd, and use confocal imaging to determine their subcellular localization. And we're excited to find that a number of the candidate proteins localized to the pirenoid, which was intriguing because for the past 25 years, the pirenoid has been thought to be primarily composed of two proteins, rubisco and rubisco activase, which is a chaperone for rubisco. In these confocal microscopy images, each of the fluorescently tagged proteins is shown in green, and the surrounding chloroplast is shown in magenta, visualized through chlorophyll autofluorescence. You can see that each of these proteins localized to a bright punctus in a hole in the chlorophyll autofluorescence, which corresponds to the pirenoid matrix. So you can imagine that we were thrilled when we started to see other proteins that also localize to the pirenoid, like this protein, which localizes to the pirenoid matrix, or other proteins that showed previously undescribed localizations, like this protein that we think localizes to the starch plates, because when we take a section through the pirenoid, we see a ring, but when we look at the surface of the pirenoid, we see what looks like starch plates with cracks in between them. And this protein, which intriguingly forms a mesh around the pirenoid, and we hypothesize fills the gaps between the starch plates. And my favorite, this protein, which forms a beautiful star-like pattern inside the pirenoid, which we think corresponds to the pirenoid tubules. We then went on to systematically do immunoprecipitations and mass spectrometry on these and other pirenoid localized proteins. And together we identified a total of about 90 pirenoid localized proteins and gained insights into their organization into protein complexes. Going forward, we're super excited because these pirenoid localized proteins now provide us and the field with molecular handles that we can use to characterize the protein composition, structure, and function of the different subcompartments of the pirenoid. The next challenge for the field is to figure out what function each of these proteins is playing in the pirenoid. The one that we understand the best is this matrix localized protein, which Luke named EPIC1 for essential pirenoid component. And we now have extensive data indicating that this EPIC1 protein serves as a molecular glue that holds the rubisco together in the pirenoid. One of the results that supports this model is based on looking at fluorescently tagged Rubisco. In wild-type cells, fluorescently tagged Rubisco forms a bright punctus where the pirenoid is. However, in a mutant lacking EPIC1, we see that the Rubisco is now found throughout the chloroplast, indicating that the EPIC1 protein is required for Rubisco's normal localization to the pirenoid matrix. In other experiments, we observed that EPIC1 binds to Rubisco, and that EPIC1 consists of multiple sequence repeats, which led us to hypothesize that EPIC1 and Rubisco each have multiple binding sites for the other, allowing the two proteins to form a dense interlocked matrix. You will notice that in this paper we drew the Rubisco arranged in a crystalline lattice. This is because, at the time, the pirenoid matrix was thought to be a crystalline solid. This view had emerged from electron micrographs taken 50 years ago, which showed crystalline arrays of particles in the pirenoid matrix of several species of algae, like this one and this one. However, Liz Freeman-Rosenzweig, a graduate student in the lab, made a series of surprising discoveries that have shown us that, at least in Chlamydomonas, the pirenoid matrix is not a crystalline solid, but rather mixes internally like a liquid droplet. One of the results that supports this view comes from fluorescence recovery after photobleaching, or FRAP experiments. Here we're looking at a cell where the pirenoid matrix is again lit up by expressing Rubisco fluorescently tagged with Venus. But now these images are false colored, with red and white being the brightest intensity. In this experiment, we took a picture of the pirenoid, and then using a laser, we bleached the fluorescence in half of the pirenoid, producing a pirenoid that is unevenly fluorescent. We then watched the pirenoids over time, and we were surprised to see that the fluorescence homogenizes on the time scale of seconds, indicating to us that the pirenoid matrix mixes like a liquid. In other experiments, we observed that the matrix can divide by fission. Here again, we're looking at Rubisco tagged with Venus in green, and the surrounding chloroplast is visualized in magenta. Under our growth conditions, the cells undergo 2 rounds of division in rapid succession. I'm going to play this movie. And you can see that the pirenoids divide by fission. They're going to divide again. And strikingly, Liz also discovered that the pirenoid partially dissolves into the surrounding chloroplast during every division. Together, these findings have led us to the conclusion that the pirenoid matrix is not a crystalline solid, but rather behaves as a phase-separated liquid-like organelle like RNA granules and nucleoli. So we had to update our model to something more liquid-like. This new view of the pirenoid as a phase-separated organelle changes how we've been thinking about its biogenesis and engineering it into plants. Now, building on our work, our collaborators Toby Wunder and Oliver Moller-Kahar showed that EPIC1 and Rubisco are sufficient to reconstitute a pirenoid matrix in vitro. Specifically, they found that mixing purified EPIC1 with purified Rubisco in a test tube produces phase-separated droplets that showed a similar mixing behavior as we had observed in vivo. But we still didn't understand how these proteins interact at a molecular level to form a liquid-like pirenoid matrix. We didn't know if the model that I'm showing you was correct, and if it was, we didn't know the identity or the number of binding sites on each protein. To address these questions, Shan He, a research scholar in the lab, sought to understand EPIC1 Rubisco interactions at a structural level. But to obtain the structure of the 2 proteins bound to each other, she could not just mix the 2 proteins together, because they form phase-separated droplets. To overcome this problem, Shan used a 2-step approach, where she would first identify EPIC1's binding sites for Rubisco, and then solve the structure of a single binding site bound to Rubisco. To identify EPIC1's binding sites for Rubisco, Shan used a peptide tiling array approach, which revealed that EPIC1 has 5 binding regions for Rubisco. In this approach, she first generated an array of peptides that tile across the sequence of full-length epic 1. In each of the spots in this array, a peptide was synthesized that corresponds to a particular fragment of epic 1. She then probed this array with purified Rubisco. We expected that Rubisco would bind to the epic 1 peptides that had binding sites for Rubisco and would not bind to the peptides that did not have binding sites. Here's a picture of one of the peptide arrays. The darker a spot is, the more Rubisco is bound to the peptide on that spot. So you can see that indeed Rubisco bound to some peptides and not to others. The order of the peptides on the array was scrambled, and when we unscramble it and plot Rubisco binding signal as a function of peptide position along epic 1, a clear pattern emerges. We observed a series of 5 binding regions at even spacings. Excitingly, the amino acid sequence of each of these binding regions was similar to the others, suggesting that all of these binding sites bind to Rubisco in a similar manner. So when we depict this as a cartoon in our model, we depict epic 1 as having 5 essentially identical binding sites for Rubisco, which are spaced pretty evenly along the sequence of epic 1. Next, Shan sought to determine where on Rubisco these epic 1 sequences bind. To find the binding sites on Rubisco, Shan synthesized a peptide representing just one binding site and mixed it at saturating concentration with Rubisco, so that all of Rubisco's binding sites were bound with this one epic 1 peptide. She then started a collaboration with HHMI's cryo-EM facility to determine a single particle cryo-electron microscopy structure of this peptide bound to Rubisco. This amazing technique uses an electron microscope to take pictures of tens of thousands of individual particles, which are then averaged together and back projected to produce the 3D structure of a protein complex. I'm showing you the actual electron density of the epic 1 Rubisco complex that resulted from this work. The Rubisco is shown in teal, and the epic 1 peptides are shown in red. We observed 8 epic 1 peptides bound to each Rubisco holoenzyme, demonstrating that Rubisco has 8 binding sites for epic 1 and providing key insights into the mechanism of binding. Now, as you remember, the epic 1 peptides in this structure represent just a portion of the epic 1 sequence. When we model the linker regions between these peptides, we obtain our current model of the pirenoid matrix, which looks like this. Here I'm showing you 3 Rubisco holoenzymes linked together by epic 1 linker proteins. Each epic 1 is an intrinsically disordered protein with 5 evenly spaced Rubisco binding sites, which bind to specific sites on the Rubisco holoenzyme, small subunits. I'm showing you an artist's rendering of a moment in time. In reality, epic 1 and Rubisco bind and unbind really rapidly, allowing the Rubiscos to flow past each other while staying together in a densely packed condensate. So in summary, we think that our discovery and characterization of this epic 1 linker protein solves the long-standing mystery of how Rubisco is held together in the pirenoid matrix. And more broadly, our studies have now made the pirenoid matrix one of the structurally best understood phase-separated condensates in cell biology. But if we zoom out, we see that we are just at the beginning of understanding how this fascinating organelle is assembled. And another major outstanding question is, given that we know that many dozens of proteins localized to the pirenoid, how are these proteins targeted to this structure? How do they know to go to the pirenoid? Moritz Mayer, another research scholar in the lab, recently made some progress on these questions when he discovered that many other pirenoid localized proteins also contain the very same Rubisco binding motifs as epic 1, such as these proteins, which have Rubisco binding motifs, and additionally have starch binding domains depicted by these brackets. are these proteins, which also have the Rubisco binding motifs, and additionally have transmembrane domains depicted by these squiggles. The prevalence of these Rubisco binding motifs among pirenoid localized proteins led us to hypothesize that these motifs target the proteins to the pirenoid. To test this hypothesis, Moritz did a key experiment which showed us that the motif is necessary to target a protein to the pirenoid. In this experiment, he started with a protein that I'm calling Cre10 here, which, when tagged with Venus, localizes to the pirenoid matrix. As you can see, it localizes to a hole in the chlorophyll autofluorescence, which corresponds to the pirenoid matrix. He then took the same protein, but mutated the Rubisco binding motif. And excitingly, he saw that this now caused the protein to localize throughout the chloroplast, demonstrating that the motif is necessary to target this protein to the pirenoid. Moritz then did another experiment which showed us that the motif is not only necessary, but also sufficient to target a protein to the pirenoid. In other words, the motif is not just important for targeting, it is actually the only thing that a chloroplast protein needs to be targeted to the pirenoid. In this experiment, he took a protein that does not natively have the motif, FDX1, which is ferredoxin, which we know localizes throughout the chloroplast stroma. He then added 3 copies of the Rubisco binding motif to this protein, and remarkably, he found that this caused the protein to relocalize to the pirenoid matrix, indicating that the motif is sufficient to target a protein to the pirenoid matrix. These findings have led us to a model for protein targeting to the pirenoid, which I will now illustrate to you by using my amazing PowerPoint animation skills. Here we have a cartoon of an algal cell with the chloroplast and the pirenoid labeled. The pirenoid consists of a condensate of Rubisco and the linker protein EPIC1, as I showed you previously. Now imagine that we have a protein with a Rubisco binding motif that starts out in the chloroplast away from the pirenoid. We expect that this protein would diffuse randomly around the chloroplast until it encounters the pirenoid matrix, where it would get trapped by binding to Rubisco. So we think that this simple mechanism explains the pirenoid targeting of many of the core pirenoid localized proteins. Now that we have a decent understanding of how the matrix is assembled and how proteins are targeted to it, another major outstanding question is, how is the matrix connected to the traversing membrane tubules and to the surrounding starch sheath? Well, it turns out that the pirenoid localized proteins that I introduced to you earlier allow us to propose a model for how these 3 subcompartments are connected. On the left is our electron micrograph, and on the right I will draw a cartoon for what we think is happening. A lot of evidence suggests that the tubules can form independently of the matrix or the starch sheath. And it turns out that 2 of the proteins that we discovered localize to the pirenoid tubules. These are the proteins that have transmembrane domains and Rubisco binding motifs. We think that these proteins link a layer of Rubisco molecules to the tubule membranes. Next, interactions of those Rubiscos with EPIC1 would allow the pirenoid matrix to coalesce around the tubules. 2 of the other proteins that we discovered localize to the periphery of the pirenoid matrix. These are the proteins that contain Rubisco binding motifs and starch binding domains, suggesting that these proteins could link the Rubisco matrix to the starch sheath. Thus, this one Rubisco binding motif could explain how the overall architecture of the pirenoid is achieved. One of the ways we'd like to test this model is we'd like to break one interface at a time by mutating the proteins that we think mediate each interface. These experiments are ongoing, but they're challenging because multiple different proteins appear to redundantly mediate each interface. Nonetheless, we've so far managed to do one experiment where instead of mutating the individual proteins that bind to Rubisco, we've instead mutated Rubisco's binding site for the motif. Since all the interactions in this model are mediated by binding of the motif to Rubisco, you would imagine that disrupting that binding site on Rubisco would break all of these interactions and would cause the whole system to fall apart, like so. Here, the Rubisco cannot bind the motif, so we would expect that even though the tubules can form, Rubisco cannot bind to them. Also, the matrix doesn't form, and the starch sheath doesn't get shaped properly. So we in fact did this experiment, and we found that indeed, mutating the motif binding site on Rubisco blows apart the pirenoid. First, I want to show you the wild type. You can see that the pirenoid has a dense spherical matrix, which is traversed by tubules, which show up as these white streaks. And the matrix is surrounded by curved starch plates. Now, when we make a point mutant in Rubisco that disrupts the binding site for the motif, we no longer can find a traditional pirenoid in the cells. Instead, all we can find are the pirenoid tubules, but they are not surrounded by a matrix, and we no longer see curved starch plates, just as expected from the model. So we're super excited at the thought that this model may very well depict how the pirenoid is assembled. We look forward to continuing to test this model with the rest of the field as we seek to understand how this fascinating structure is built and operates. And more broadly, we hope that this motif principle may provide insights into the organization of other phase-separated organelles across biology. To us, this really seems like a fascinating example of how an organelle that at first appears quite complicated, with its 3 different subcompartments and complex geometry, can actually be assembled based on very simple principles. We hope that our basic advances in our understanding of how the pirenoid is assembled will also pave the way for ultimately engineering this structure into land plants. With that goal in mind, our collaborators Nikki Atkinson and Alastair McCormick have started to transfer the components that we're discovering into the model land plant Arabidopsis. And very excitingly, they've actually recently been able to reconstitute the clammy pirenoid matrix in a land plant. First, I'm going to show you a wild-type Arabidopsis plant, chloroplast. In this plant, the Rubisco is localized throughout the chloroplast. Nikki and Alastair then expressed EPIC1 and modified the Rubisco so that it can bind to EPIC1. And super excitingly, they see that when they do this, the engineered chloroplasts now contain a large condensate of Rubisco, which they showed mixes internally, as we've seen for the matrix and clammy. We are super excited by this early success and look forward to working with them and with the rest of the field to hopefully complete the engineering of a functional pirenoid in land plants. So what is the next big challenge? Well, these pirenoid matrices currently don't really do anything for the plant, and we wouldn't expect them to, because for the pirenoid to work, we need to feed it with concentrated CO2. So in the last few minutes of this talk, I'd like to talk about how we think the pirenoid is fed with concentrated CO2 and how we plan to engineer this into plants. In the cartoon that I showed you previously, I greatly oversimplified how CO2 is concentrated by showing you this magical arrow. And here, I want to explain in more detail how we think this CO2 concentration works. First, I will get rid of the plasma membrane, because we think that most of the CO2 concentrating activity happens at the level of the chloroplast. Next, I want to draw a more detailed cartoon of the inside of the chloroplast. Here again, we have the chloroplast envelope, which separates the cytosol from the stroma. And now inside the stroma, I have additionally drawn the thylakoid membranes, which contain an additional compartment, the thylakoid lumen. As I showed you in electron micrographs, the Rubisco matrix is actually not clustered by itself, but rather surrounds a portion of the thylakoid membranes, which form the tubules that traverse the matrix. And the matrix is surrounded by a starch sheath. Together, these three subcompartments form the pirenoid. Now, the model I'm going to show you is based on ideas from many researchers in the field. And these different groups have all proposed slightly different variants of the model, and the one that I'll show you is the simplest one that we can make by incorporating their ideas. In this model, algae concentrate CO2 using two kinds of proteins, carbonic anhydrases and bicarbonate channels. Now I'll walk you through how the CO2 goes from a low concentration outside the cell to a high concentration inside the pirenoid. We think that the CO2 diffuses across chloroplast envelope to the stroma. There, it's converted by a carbonic anhydrase to a high concentration of bicarbonate, which crosses the thylakoid membrane via a bicarbonate channel. The bicarbonate then diffuses along the thylakoid lumen into the pirenoid tubules, where another carbonic anhydrase converts it back into CO2. The CO2 then diffuses back across the thylakoid membrane and into the pirenoid matrix, where it is captured by Rubisco. Some of this CO2 leaks out of the pirenoid and then is recaptured by the carbonic anhydrase in the stroma. You may wonder how the system concentrates CO2 at all, given that all that we're doing is converting CO2 to bicarbonate and back. The key is that the equilibrium ratio between CO2 and bicarbonate is highly dependent on the pH, and the two carbonic anhydrase reactions happen at different pH. The first conversion happens in the stroma, which is around pH 8, which strongly favors a high concentration of bicarbonate, whereas the second conversion happens in the thylakoid lumen, which is at pH 6, which favors approximately an equal concentration of CO2 and bicarbonate. And thus, chaining these two conversions in sequence results in an increased concentration of CO2 in the pirenoid relative to in the stroma. Now, this is just a model, and if we're going to put a lot of effort into engineering a pirenoid, we'd really like to be sure that this system actually can work. So we started a wonderful collaboration spearheaded by two students here at Princeton, Alexandra Wilson and Chen-Yi Fei, in collaboration with two biophysicists, Ned Wingreen, who is another professor at Princeton, and Neil Mangan, who's a professor at Northwestern University. And together, we tested whether such a CO2 concentrating mechanism actually works by creating a reaction diffusion computational model. Basically, it's a three-dimensional model where metabolites like CO2 and bicarbonate can undergo reactions, cross membranes, and can diffuse around. And very excitingly, this model worked. And the CO2 concentrating mechanism works in silico. And in fact, the thicknesses of the arrows that I drew here represent the approximate fluxes that we observe in the model. And the size of the letters represents the approximate concentrations of CO2 and bicarbonate in the model. The model predicts a 7-fold increase in Rubisco activity. We observe that under air levels of external CO2, the mechanism can be entirely driven by pH differences and passive transport with no need for active transmembrane pumps. And the mechanism is energetically efficient, consuming only about 1.5 ATP equivalents per CO2 fixed in air, which is only about 10% of the energy that it takes to fix one CO2 using the Calvin-Benson cycle. But most exciting of all, I think, is just that the system works. There are no mystery components, there's no magic. We know the identities of the carbonic anhydrases and the transporters. So this work personally makes me very optimistic that, guided by this computational model, we will be able to engineer plant cells to supply concentrated CO2 to their Rubisco matrix. So, in summary, I've told you today that the algopirenoid enhances carbon fixation. But when we started 11 years ago, we knew almost nothing about it at a molecular level. Because there were very few genetic resources available in algae at the time, we developed the world's first genome-wide collection of algal mutants with known gene disruptions, and used this collection to identify mutants with defects in the pirenoid. We then used a systematic localization strategy to identify even more components and to gain insights into the anatomy of the pirenoid. I highlighted our discovery that the pirenoid is a phase-separated liquid-like organelle and not a crystalline solid as had long been thought. Then I told you about our molecular studies, where we discovered a key linker protein that holds the rubisco together in the pirenoid. And I shared our discovery that many pirenoid proteins contain the same rubisco binding motif, which mediates their targeting to the pirenoid, and also suggests an elegant model for how the matrix is assembled onto the tubules and how the starch is assembled onto the matrix. We hope that our efforts will ultimately allow engineering of a pirenoid into land plants to improve crop yields, and in fact we're making good progress on this with our collaborators. So finally, I'd like to thank the members of my lab who did the work that I presented to you today, both the current and past members, and we're very grateful to our wonderful collaborators and to our funding sources. I also want to mention that we have a postdoc position available. Thank you for listening, and I look forward to your questions.
Thank you very much, Martin. Very nice presentation and interesting